Good morning, Don. Good morning, Kathryn. My name is Irina Ellison. I'm the moderator for this session, and we'll get started in just about 10 minutes as we're waiting for the other presenters. Good morning, Megan. My name is Irina Ellison. I'm the moderator for the session, and we'll get started in just about 10 minutes or so.
Okay, good morning.
Good morning. Good morning, Ariana. I'm Irina Ellison. I'm the moderator for this session, and we're going to get started in around 8-9 minutes or so. We'll try to start as close to on time as possible. We just are waiting, I think, for one more presenter. Good morning, Chris. We're just waiting on one more presenter and just waiting until 9 as well for us to get started.
All right. Well, good luck, everybody.
Thank you. Good morning. I think we have all of our presenters here now. We'll just wait 2 more minutes for 9 o'clock and then we'll get started. And we're going to go just in the order of the program. So Megan, Kathryn, Ariana, and I don't want to say your name wrong. Is it Iyinyeoluwa?
Yes.
I tried. I'm sorry. I'm not.
You're good.
Okay. So we'll just wait another 2 minutes or so because we may have some more folks that want to listen to your presentation. So we'll just wait a few more minutes and then we'll get started. All right, we're going to get started. Good morning, everybody. Happy Sunday. Happy conference. My name is Irina Ellison, and I'm the moderator for this session. I'm going to introduce all of our presenters and their topics first, and then we will go through according to the order in the program. So our first presenter is Megan Anderson from the University of Hartford, and she is going to be presenting on how do cells become cancerous, determining the effect of p53 in daughter cells during prolonged prometaphase. Our next presenter will be Kathryn Bosley from SUNY Cobleskill, and her presentation is The Impossible Cow, Hornless and Productive. Our third presenter is Ariana Gazaferi from the University of St. Joseph, and her topic is synthesis and testing of sulfonamides as bacteriostatic compounds. And last but not least is Iyinyeoluwa Okulate from the University of Maryland Eastern Shore, and her presentation is functional characterization of insulin receptor in zebrafish kidney. And our panel this morning is on applied genetics and biochemistry. By training, I'm a biologist, so I'm very much looking forward to all of your presentations, and I'm going to turn it over to Megan.
All right, thank you, and good morning, everyone. I'm going to share my screen. Let's see here. I'll get this into present mode. Okay, is that good? Can everyone see it?
Yes, we can see it.
Awesome. So as mentioned, my name is Megan Anderson, and I'm from the University of Hartford. And over the past 2 semesters, I have had the pleasure of working with my faculty advisor and mentor, Dr. Iyinyeoluwa, to determine the effect of p53 on daughter cells during prolonged prometaphase. So let's see. Okay. Just a little overview of the project. We looked at cancer cells during this, as the hallmarks of cancer cells have been well characterized, but there's still a lot that is unknown about how cells can become cancerous. However, there's a group of proteins called tumor suppressor proteins that have shed some light on this, and more specifically the protein p53, as it is defective in over half of human cancer tumors. And there's some previous research that has shown that if daughter cells go into this prolonged arrest during prometaphase, then once they're released from arrest and go through the rest of mitosis, the daughter cells will subsequently arrest in G1. And it has been proposed that p53 has been involved in this arrest, and so that is what this project is studying. We used 3 different cell lines and then treated them all with a cell synchronizing drug called RO3306 to synchronize them at the G2M phase. And then they were released and treated with nicotazole for either 1.5 or 2.5 hours, which is a microtubule destabilizing drug. And after their treatment, they were subsequently harvested and analyzed via flow cytometry and Western blot analysis. So what does this all kind of mean? Well, the purpose was to determine if P53 is activated in response to cell cycle damage sustained during this prolonged prometaphase. Once cells grow and divide into 2 daughter cells, they have to go through a process called the cell cycle. And this is broken up into 4 stages, the first being G1, where the cell grows, second being G2, where all the DNA is replicated, second being S phase, where all the DNA is replicated, and then the third being G2, where the cell again grows and prepares to divide in mitosis, which results in 2 identical daughter cells. And in order for this to go on smoothly and to ensure that the cell does not experience any damage that could then damage other cells later on, there are checkpoints involved. This first checkpoint is at G1, and this is where the cell ensures that it's grown enough and that it's in the proper environment. The second checkpoint is in G2, where the cell again ensures that it's grown enough and is in proper environment, but it also needs to make sure that all of its DNA has been replicated. And then lastly, there's a checkpoint at the metaphase-anaphase transition in mitosis. And this is where this metaphase-anaphase transition is where the cell needs to ensure that all of its chromosomes are aligned at the center of that metaphase plate and that they're attached to microtubules, which are these string-like projections. If this is not the case, then this cell will go into prometaphase, as depicted here, and it will arrest in prometaphase until this problem is resolved. Now, since P53 has been known to be involved in other cell cycle checkpoints, like in the G1 and G2 checkpoint, it is plausible and it's been proposed that P53 could be involved in this mitosis and mitotic checkpoint. So P53 is, as mentioned, a tumor suppressor protein, and it is always present in cells unless it gets degraded, and it usually gets degraded unless it's activated. When it's activated, it is activated in response to DNA damage, and then it will go on and activate transcription of other genes such as P53. In order to do this, it needs to form tetramers, which are these little 4 subunits of P53. And then it can induce those transcription of other genes and then induce cell cycle arrest and apoptosis, where the cell will die. So the previous research that led to this experiment was that researchers looked at cells being arrested in prometaphase for over 1.5 hours. And they found that the daughter cells of those that were arrested in prometaphase for over 1.5 hours subsequently arrested in G1, and they deemed this to be a prolonged prometaphase. They also saw that P53 was activated in response to this prolonged prometaphase. However, if cells were arrested in prometaphase for less than 1.5 hours, it was seen that they would just proliferate normally. Therefore, this project is trying to look at cells that have P53 and also cell lines that lack P53 or lack domains that will allow P53 to go on and induce transcription of other genes. We also were just trying to mimic the results of the previous research just to ensure that P53 is in fact activated in response to a prolonged prometaphase, which is why we decided to do a 1.5-hour nicotazole treatment group. This served as that treatment group that was less than 1.5 hours. So it was expected that these cells would proliferate normally, as well as a 2.5-hour treatment group, which was over the 1.5-hour mark. So this was intended to mimic that prolonged prometaphase. The cell lines used was MCF10A cells, which have wild type P53 or normal functioning P53. The MCF10A cells with delta P53, which express an shRNA that will target that p53 protein's mRNA for degradation, meaning p53 is non-functional. And then lastly, MCF10A cells that overexpress the oligomerization domain, which interferes with p53's ability to form those tetramers and go on and induce transcription of other genes such as p21. And this image to the left is of the MCF10A cells prior to harvesting. These are all cells that were treated with nicotazole. The methods of analysis was flow cytometry, which determines the cell cycle distribution and the percent of cells in each stage by analyzing how much DNA is in the cell. So this image over here to the left shows kind of what the normal untreated, unsynchronized cells would look like. In G1, there's only one copy of DNA, and so there's a big peak here as G1 is longer. And then once we get to G2, there's two arbitrary units of DNA as the DNA has already been replicated. And so there's a little smaller peak here as G2 is not as long. The second method of analysis was Western blot analysis, and this determined the level of different cell cycle proteins within the cells. And we looked at a few different proteins, but specifically we're looking at p53, p21. So as mentioned, it was expected that there would be p53 activation in response to this prolonged prometaphase. So we expected that the MCF10A cell treatment with 2.5 hours of nicotazole, we would subsequently see these cells being arrested in G1 and then have an activation of p53. It was also expected that the MCF10A OD cells would show activation of p53 in response to the 2.5 hour treatment group, as these cells can activate p53, they just cannot further activate other cell cycle arrest proteins such as p21. However, the MCF10A cells with delta p53 should not have shown any p53 activation, as their p53 mRNA gets degraded. And what we saw was a tad unexpected. So I'm just going to quick explain these images up to the right. For all cell lines, there were 2 samples of untreated cells taken and 2 samples of RO3306, so the synchronizing drug, samples taken. Up to the right, the top image is the 1.5 hour treatment of nicotazole for MCF10A cells, and the bottom is the 2.5 hour treatment. So we have the untreated cells, and then RO3306, and then cells harvested immediately after release from nicotazole, so that nicotazole control. And then cells harvested 2 hours, 4 hours, 8, 12, 18, and 24 hours after. And what we saw initially with these untreated cells was expected. There was about 53% in G1 showing normal cell proliferation. And then we got to the RO3306 cells, and there showed about 50% or less in this G2M phase, which was a little unexpected because since they should be synchronized, we should have seen a higher percent of cells here. After release from nicotazole, it showed that about 2 hours after, for both treatment groups, is when cells returned to G1 and we saw some arrest. In the 2.5 hour treatment group, there was 73% of cells in G1 2 hours after nicotazole release, compared to only 58 in the 1.5 hour treatment group. So this suggests that there was more G1 arrest in that 2.5 hour treatment group, which was likely due to that prolonged prometaphase as expected. However, you can see that from the 2 hours to 24 hours in both treatment groups, the percent of cells in G1, so 58, and then drops to 50, and in 2.5 hours, it goes from 73 to 60. This drop in number suggests that they were not fully arrested, because if cells were fully arrested, then this number would stay basically the same. The Western blot analysis of these cells showed that p53 was activated in response to that prolonged prometaphase, so that 2.5 hour treatment group over here, image B. But then it also showed that there was p53 activation in the 1.5 hour treatment group, so in image A, which was not expected. What was also kind of odd was that in these untreated cells, so this first column here of both treatment groups, there showed p53 activation. And since these cells did not have any damage, there should not have been any p53 activated, which led us to believe that this Western blot analysis should be repeated. We also saw that p21 was activated in response to p53, which was expected, and that p21 activation showed up at around 0 hours, so right after the cortisol release, as expected in the 1.5 hour treatment group. And then it was a little more constant throughout the 2.5 hour treatment group. Again, these discrepancies led us to believe that the Western blot analysis should be repeated. For the MCF10A cells with delta p53, some similar results in the flow cytometry was seen as compared to the 10A cells. However, just in lesser numbers, we saw the normal amounts of cells in G1 for untreated cells, about 41%. But then the other samples saw 49% of cells in sub G0, which means that these cells were dead or dying. And so that was kind of a conflicting result that we are unsure about. We again saw that after 2 hours following the cortisol release, most of the cells went back into G1, and then this number dropped over the course of the 24 hours, suggesting that there was not a complete G1 arrest. The results of Western blot analysis were consistent for p53, as there was just very minimal basal levels of p53 seen across both cell lines as expected. However, we did see some presence of p21, this bottom row here, which was unexpected, as since p53 is degraded, it should not be able to go on and induce the transcription of p21. The only explanation we had was that since there was presence of this phosphorylated form of p53, the phosphoser20, this could have induced p21 transcription. But again, in the untreated cells, we saw the presence of this, which should not have been the case, as these were not treated with anything. For the MCF10A OD cells, we saw the untreated cells both had a large percent in G1 as normal, but then we saw that both treatment groups remained in this G2M phase for a lot longer period of time, almost up to 8 hours following the cortisol release, before they subsequently went back into G1 and showed relatively normal cell proliferation. The other thing that was interesting about the OD cells was that there was a lot more cells in sub G0, meaning that there was a lot more cells that were dead and dying, or apoptotic cells. The Western blot analysis of the OD cells showed that p53 was activated in response to both the 1.5 hour treatment of the cortisol and 2.5 hour, which was unexpected, as we only thought it would be activated in response to 2.5 hour treatment. And then we also saw activation of p21 in that 2.5 hour treatment group, which was again unexpected, as these cells do not express the domain that allows p53 to form tetramers and induce transcription of other genes. And again, in these images, there's discrepancies between the untreated cells for both images A and B, and same with the RO3306 treated cells, which led us to believe that Western blot analysis of some of these proteins needs to be repeated. So overall, it was a success in that we did see that p53 was activated in response to the prolonged Prometaphase. However, we also saw that it was activated in response to that 1.5 hour Prometaphase arrest. And so that led us to believe that we need to do some more manipulating of the treatment of nicotazole to really be sure that cells are in a prolonged Prometaphase arrest. So we thought that if we were to repeat this experiment, we could do a 1 or 1.5 hour nicotazole treatment, as well as a 3 hour nicotazole treatment, just to ensure that there is a prolonged Prometaphase arrest there. And we also wanted to treat the cells with the cell synchronizing drug for about 30 minutes longer. We did a 1 hour for this experiment, but as the flow cytometry results showed, it was not clearly, they were not all synchronized, and so this could have been a source of error. Lastly, we wanted to rerun the Western blot analysis, which in a normal year would have been done, but due to this like opening and closures of school and myself being in and out of quarantines and stuff and just all the COVID protocols, that wasn't possible. But that's what we hope to do if we were to continue this. So that was the completion of my presentation. I would just like to thank my advisor, Dr. Levesque, for really all of her work. Even these past 4 years, I started research with her when I was a freshman. And then also Dr. Oakes and Dr. Jones for all of their help and support in the honors program.
Great. Thank you so much, Megan. I know we have a couple questions in the chat, but we'll hold on to those for the end. Our next presenter is Kathryn. I'm going to pass it along to you, Kathryn.
Excellent. It's for some reason not letting me show it.
We can see it when you go and we see that you're pulling it up. And then I think if you just click on the leftmost button from presenting. Because we see that you're screen sharing. If you go all the way to the left from beginning, I think that should allow you. Yep, where you were. Yeah. There you go. Perfect.
All right, so my presentation is on the impossible cow, hornless and productive. And I'm Kathryn Bosley from SUNY Cobleskill. And quick outline, I'm going to present some background information, my materials and methods, results discussion, conclusions, recommendations, as well as references, acknowledgements as well. So why hornless cows? So in the wild, horns are useful for cattle. However, in modern production systems, not so much. So horns increase the risk of injuries, which can lead to infections and lower carcass qualities and economic losses, as well as human safety concerns. So in response to this, it's common practice in the dairy industry to disbud or dehorn. Disbudding occurs before 2 months of age, before the horn tissue has formed with the frontal sinus, and this procedure is minimally invasive, especially with pain management. However, dehorning, which is after 2 months of age, occurs after the frontal sinus has already merged with the horn core, and this procedure to dehorn has a greater risk of infection. So pulled, which is naturally hornless genetics, saves the time, money, and stress that would come from dehorning. The American Veterinary Medical Association cites dehorning as an area of concern for animal welfare. So dehorning costs $6 to $25 per head, while pulled genetics costs an additional $0 to $26 per head over traditional horned sires, with greater over for the heterozygous pulled bulls rather than homozygous pulled bulls. So the break-even point per head would be almost $12 for homozygous pulled bulls and almost $6 for heterozygous pulled bulls. Some disadvantages of pulled genetics is that there is a limited availability of pulled sires. So between 2008 and 2018, only 5% of artificial insemination sires available in the US for the Holstein, Jersey, and Brown Swiss breeds were pulled. And these bulls actually lagged 20 to 30% in net merit behind their horned counterparts, although they showed the same genetic gain rate when used in a herd. As well as there's also difficulties with mixed management for having both polled and horned calves, because a lot of disbudding occurs before the horn bud is evident. So you could potentially disbud all your calves, even the polled calves, which would minimize any advantage you would get from using polled genetics for animal welfare or time and management costs. There's some concern on inbreeding, but it has been shown so far that there is not an increased risk of inbreeding for using polled genetics versus the horned counterparts. There's also the concern of horn lobbying groups that are pushing against polled genetics because they believe horns are more natural. So some strategies for implementing polled genetics. So it has been shown that you can use genetic engineering, specifically CRISPR, to actually insert polled genetics into traditional horned genetic lines. However, this is severely limited right now by government regulations as well as anti-GMO lobbying. The more traditional method of integrating polled genetics is to use gradual selective breeding, and since the polled condition is an autosomal dominant, a polled calf can appear within the first generation. For using heterozygous polled bulls on an entirely horned herd, about half the offspring would be polled. Using homozygous polled bulls, you would have all of the offspring be polled. However, the genetic merit is considerably lower for the homozygous polled bulls versus the heterozygous, and both are lower than the horned genetic sires, as mentioned before. Another possible strategy for implementing polled genetics is crossbreeding with predominantly polled breeds. A lot of red breeds are used, like Norwegian Red or Montbéliard, which have the added advantage of higher fertility rates than traditional Holstein sires. So my research study's objective was to evaluate strategies for practical application of selectively breeding polled genetics into a horned Holstein herd. I specifically used my campus's Holstein herd. So my materials, so the cow group were all of the Holstein cows that were coded as okay to breed. And my bull groups, so the control group was all of the US bulls from the December 2020 top 200 genomic TPI list. TPI is Total Performance Index, which is a composite selective index that is the main one used for the Holstein breed in the United States and is set every year by Holstein Association USA. My 3 experimental groups were my homozygous polled group, the PP, heterozygous polled, PC, and observed polled. Observed polled were not genetically tested, so it's unknown whether they were homozygous versus heterozygous, and were simply coded as PO within the Holstein Association's genetic database. So my methods, I imported the cow data from Dairy Comp 305, which is the herd's management software, which contains all of the data from all of the cows. And this data was imported into Redbook Plus Multimate, which is a desktop software program distributed by Holstein Association USA. The version I used was the December 2020 version, which was the most recent version at the time of the study. The bull groups were created using the Redbook Plus function in Redbook Plus Multimate, and the TPIs between all the groups were compared. The mating goals for the herd included breed average production, breed average linear traits with an economic weighting, as well as a 60-40 ratio for types of conformation traits versus production traits. So these mating goals were implemented into the Multimate part of the software program to create this mating index. So the mating index indicates the genetic merit within the herd. So it is only specific to this herd, and these mating goals, this index does not, it just simply ranks them within the herd. It is not applicable outside of this herd. And the matings were run using the multi-mate program. So each bull group was mated against the same group of cows, and the mating index for the first, second, and third choice bulls that the program chose were recorded, and these were analyzed using Microsoft Excel. And using Microsoft Excel, I calculated the significance between the different groups for mating index using a two-tailed t-test. So as far as results, comparing the different TPIs of the bull groups, the control group was higher than a top 10% of the average TPIs for all bulls within the USA. The 3 experimental groups were similar for TPI to an average bull. As far as which would increase polledness the fastest, has been shown that the homozygous polled bulls would increase this quicker than observed, then the polled carriers, and then control, which contains only horned bulls. So from most to least effective for increasing polledness. As far as increasing TPI, the control group had the highest TPI, then PC, then homozygous, and then polled observed. However, how does this compare for the herd mating goals? So as far as the herd mating goals, the highest mating index was seen for mating the herd to the first choice control bulls, second highest for the second choice control bulls, and the third highest for the first choice polled carrier bulls, so the heterozygous bulls. However, when comparing these groups between each other using the t-tests with a significant value of p being less than 0.01, the control group and the polled carrier group were determined to have no significant difference between the groups as far as mating index, while a significant difference was seen between mating versus the controlled and the homozygous, or the controlled and the observed polled. So since there was no significant difference between the control group and the polled carrier group, this means that the polled carrier group is as effective at meeting this herd's mating goals as using any of the controlled bulls, with the added advantage of half of the offspring being polled. My conclusions and recommendations: for this herd, the best strategy for implementing polled genetics would be the use of the polled carrier bulls. These bulls are similar enough as far as furthering the herd's mating goals to the control group that they would be able to implement polled genetics without sacrificing too much on genetic merit. Determining the best polled breeding strategy may depend on the herd's breeding goals, and some limitations of my study was that we only focused on one generation, used a computer model, and only used Holstein cattle. For further research, different mating goals could be used in different herds, as well as other breeds such as Jerseys could be used. These are some of my references. And I would like to acknowledge my college, as well as my advisor, the farm manager, and the dairy manager.
Great. Thank you so much, Kathryn. Wonderful presentation.
Thank you.
Our next presenter is Ariana.
All right, hi everyone. Let me just share my screen. Let's see. All right, so it should, there we go, okay. All righty. So the research that I did happened last semester. So the topic is the synthesis and testing of sulfonamides as bacteriostatic compounds. So just a brief kind of overview about what this project was all about. So recently, especially in the times that we've been experiencing with the COVID-19 pandemic, there has been the strong need for scientists, for researchers, and all these other healthcare professionals like doctors, nurses, pharmacists, etc. And so oftentimes, a lot of students who want to pursue these types of fields in research or in medicine and things, sometimes they don't know about how even the whole process of like a drug discovery and how these medicines work and how they were first kind of developed are made. And so this was kind of brought up many times, and I've also noticed this thing as well, especially someone as someone who wants to go into such a research field like that. And so the purpose kind of was the creation of this drug discovery lab. And so it utilizes both organic chemistry and biology just to kind of help students be aware of the fact that how these medicines are created and then how they are tested and implemented so that they can be used in the future and on people. So we made different compounds using this organic chemistry synthesis, and then they were able to be tested on bacteria samples to see if they truly were bacteriostatic. So just some background about how antibiotics and antimicrobials were kind of discovered. So one of the main leaders was Alexander Fleming, and he was known for the creation of penicillin, which is an antibiotic, one of the first antibiotics kind of discovered. And so he found this by kind of accidentally through a contaminated plate of staphylococcus with mold. And he found that mold created this kind of mold juice, and that inhibited kind of the growth of staphylococci. And so that was later then he found out that was actually one of the group for penicillin. Another main leader was Paul Ehrlich. He discovered something called the magic bullet. And it was interesting enough that he found that it led to like chemotherapy a lot. And so the magic bullet is something that can kill the thing that is infected or the bacteria and the cells with bacteria, but not the actual host. And that was later used for things like chemotherapy. And so in doing this, he also used compounds called anilins and synthetic dyes to kind of develop this idea. And eventually he also developed an antibiotic called subarsin, and that was used for treatment for syphilis, which was very prominent during when he was kind of doing all this research on it. And then so now this kind of leads to the idea of just antibacterials in general. So antibacterials are something that can prevent the growth of bacteria. So they can do this different ways. So they can either stop the bacteria from replicating in the body, or it can kind of change the coding in the DNA of the bacteria. And so there's these two categories. So there's bactericidal and bacteriostatic. So the main focus of my research was on bacteriostatic. So bactericidal is something that completely kills off the bacteria. So that is an example of antibiotics. But bacteriostatic, which is what I looked at, just prevents the growth of bacteria and does not completely kill it off. And so this can actually bring about some use in modern world sometimes if you don't want to completely kill off like the microbiome kind of in your body and stuff. It's helpful to use bacteriostatic drugs. So there's many different advantages for choosing bacteriostatic versus bactericidal. And there are some various different types of drug classes for bacteriostatic, but my main focus was the very bottom one, which is sulfonamides. And so to the left is a picture of an example of a sulfonamide, which is sulfomethoxazole, which is commonly used for many bacteriostatic ways. And so in sulfonamides specifically, they work on the folate synthesis of bacteria. And so folic acid is needed as a metabolizer for bacteria in order to kind of reproduce and grow. And so in order for folic acid to be synthesized, it has to cross over into bacterial cells. And that's in the cells, that's when they're able to be grown and for bacterial growth. And so in order to get across the membrane for bacteria, they use something called PABA, which is para-aminobenzoic acid. And so interesting enough, which I'll show in the next slide, a comparison between PABA and sulfonamides. Sulfonamides kind of mimic PABA and they fit where PABA usually goes in. And so thus sulfonamides can inhibit the folic acid growth and thus bacterial growth. And so, yeah, so to the left, that is a common sulfonamide, so sulfomethoxazole trimethylprim. And so here is just a comparison of PABA and sulfonamides. So to the left is PABA. So up here, this para-aminobenzoic acid this right here is the functional group carboxylic acid. And so if you look to the right, this is the comparison of the sulfonamides. So this SO2 NH2 part is perfectly mimics this carboxylic acid, interestingly enough. And that is how then, by using sulfonamides instead of CABA, we are able to inhibit that folic acid growth, and then the bacteria will not be able to replicate and synthesize. So the basic experimental process. So the first part, again, as previously mentioned, was kind of the organic chemistry part. And so here I just kind of showed it was done in two parts. So in the first part, we started off with acetobenzosulfonyl chloride, and we would reflux it with a chosen amine. So the R stands for something attached to this NH2 group. And the NH2 is what is classified as the amine group. And so in this whole research, there were 4 different amines that were chosen. So 2 of them that were previously done last year with 2 other students, and then 2 new ones. And so we would reflux the amine with the acetobenzosulfonyl chloride, and we would also then add acetonitrile and potassium carbonate. And so that was refluxed for about 30 minutes. After reflux, the solution then kind of cooled to room temperature, and we poured it into ice water. And so after doing so, then HCl was added in small concentrations until we obtained a certain pH of around 2 or 3, so pretty acidic. And then after that, we would, upon adding the HCl, sometimes a solid would actually start forming. And so we were able to collect that solid out of the ice water and with vacuum filtration. We would leave it for like a day or so just to dry out. And that actually turned out to be the intermediate of the product, which is shown in this top right corner right there. And again, the R group is whatever that was attached to the NH2 in our amine. And then the next day, we would be the second part of this synthesis. So we would take that intermediate and we would reflux it with HCl for a longer time, for 60 minutes. And then we would let it cool to room temperature. Then we would put it in an ice water bath again. And instead of adding HCl, then we would neutralize it with potassium, not sodium bicarbonate, until we reached again a certain pH of about 7 or 8. So we wanted to keep it pretty neutral. And after adding that potassium, the sodium bicarbonate, then we would, a solid actually would form out again, and that would be the final product, which is seen in the bottom right-hand corner. So it was pretty neat stuff, I would have to say. So for the first experiment, we used an amine called aniline. And so again, this is just showing the process of how that NH2 is kind of attached on where that sulfonyl chloride and the CO2Cl part. And so we would make this final product using that previous description of the process that was talked about before. And so this was one of the first compounds that was used previously. So it was good to just kind of do it again and repeat what the other students had done, just to kind of as a reference and make sure that it's confirming what they originally did as well. The second one, we used 1-phenylethylamine. And so again, this was another one of the compounds that was previously done before. So again, this is just showing the same process of how the NH2 is attached on the SO2Cl part of the benzyl sulfonyl chloride. And it's just, again, whatever results we get was just kind of confirming to see what the previous students got and if they matched kind of similarly. Experiments 3 and 4 are going to be the kind of the new ones that we haven't tested yet, and they look a little bit different than how the previous ones were done. So the other ones were more of a ring or aromatic ring type structure, and these were more chains. So it was interesting kind of difference between how, if a solid was able to form out right or perfectly, just as how like the previous 2 were done. So for this one, we used 3-aminopropanol. And so again, this is just showing how that chain is added onto the benzyl sulfonyl chloride to make the final sulfonamide product. And then the fourth and last one that we did was 3-methoxypropan-1-amine. And so as you can see here again, we used a chain instead of in a ring type structure. And so that also, which I'll talk about a little bit later, also altered how that product was able to be formed, both the intermediate and the final one, which was really interesting to see and compare to compared to experiments 1 and 2. So once those compounds were all made, then it was time to kind of set up for bacteria testing. So the first thing to do was to make the concentrations of each sample that were going to be used to test on the bacteria. So we aim to make about 10 milligram per ml ethanol concentrations for each thing. So we would take about 10 milligrams of each of the final products from the experiments, and then use 1 ml of ethanol and kind of swirl around and make sure that the product fully dissolves into the solution. If it didn't, then we would just kind of put that little vial in like a hot water bath, and it was able to dissolve completely in there. So I listed for each of the experiments what the concentrations ended up being. So as you can see here, it's not perfect, as long as we got roughly around there. But again, that might have affected the future results, which will be talked about again. And so we used both E. coli and E. aerogenes bacteria. Previously done, they used just E. coli. So I wanted to see kind of if we used also something that was a different bacteria, also the same kind of gram status as well. And also it was interesting because E. aerogenes is known to be kind of, has a lot of drug resistance to it. So it was interesting to see if anything was possible, if maybe this type of bacteria would cause drug resistance with maybe some of the concentrations and not show a different bacteriostatic effects as we hoped. So 5 plates were each used for both of the types of bacteria. And then we had 1 plate that was just as a control, which was just the ethanol. All right. So now we get into the results. So the first part, we wanted to confirm that what we made for the actual chemistry process was correct, that we made the correct intermediate and the correct final product, because if we didn't, then it would not be a true sulfonamide, which is what we hoped. So what we did for the chemistry is what called an HNMR, where basically you run a sample through a machine and it tells you where the certain hydrogens are in the compound and which carbon it's attached to, and that can help you kind of construct what your final product should look like. So if we take the final product from experiment 1, and so I kind of showed in different colors where each of the hydrogens were that showed up on the NMR. And so we can see that it did turn out to be a success, and that all the important hydrogens that we wanted to notice were found on there. And this confirms what the previous students had done as well, and their results that we got. So the chemistry part for at least the first experiment was correct. And so in the red, we can see like the aromatic hydrogens. Also in the blue, we can kind of see them around the same area, which is typically in the range that they should show up in, between 6 to 8 ppm. And then we have this NH hydrogen that's all the way to the very end, around 10, which again is expected where it should show up. And then we have the NH2 again showing up around 7 to 7.2, which is the range that it should have been in. A lot of this stuff around 0 is the solvent that we use to kind of dissolve it to get it ready for the HNMR, so TMS. And so that always shows up as 0, kind of just like as a benchmarker for it. So experiment 2 was also determined to be a success as well, at least for the chemistry part. We can see that we have the CH3 in purple, which is should be around 1 to 3 ppm. So it shows up exactly where we would want it to be. We have the aromatic hydrogens again showing up in that same area to determine that it was a success as well. And the NH also was super important to kind of determine, because that NH, in order to determine if the synthesis was successful, that NH had to be attached on there. And so finding that NH around a high ppm also confirmed that the correct product was made. So again, experiment 3 also determined to be a success. This one was a little bit longer, had a bit of a longer chain than the other ones. So it showed up definitely a lot more hydrogens and a little bit more neater as the synthesis kind of went on, and the correct way to kind of synthesize things was perfected a little bit more. And so the aromatic hydrogens showed up in the same areas around between 6 and 8 ppm. That NH, again, we were able to find it. It's very typically the NH is very variable in where it shows up, but it's consistently been showing up, which is sometimes very rare, but really good that we were able to find it on there. And then again, some of the hydrogens that are attached to a lot of the carbon peaks in that long chain showed up around between 1 and 3 ppm, which is where they typically should be expected. And so then again, this confirms that the correct product was made, at least for the chemistry part. And then finally, again, the same thing occurred for our fourth experiment. The aromatic hydrogens showed up around the same area. A lot of the same carbon, the CH2 peaks also between 1 and 3 showed up as well. We have the CH3 that's attached in the ether also showing up, and it's a very strong peak as well. So that also then again shows that correct products made, as well as the NH, again, confirming these ones. So experiment 3 and 4 were definitely a success as far as being like new products to synthesize into. And then experiments 1 and 2 were also a success and kind of confirmed what the previous students had done as well. So then we come into the bacteria testing. So after plating them and things, so we used a Kirby-Bauer assay where you would take little filter discs and then you would dip them into the different concentrations that we make. And then we put them on the plated bacteria, on each of the plates where the bacteria were kind of made a lawn over. And then you would incubate them for about 18 to 24 hours. So we did that one day and then we let them incubate for the next day. And then we came back on the same day, the next day to see if what the results were. And so what is expected is you want to see for a bacteriostatic compound, you want to see a zone of inhibition, which shows that it will look like a little ring around each of the filter discs, and it will show and see that in that little area, you can then measure it and see if bacterial growth there was inhibited and by how much.
Ariana, I just want to, sorry to interrupt, but you're beyond 15 minutes, so you just need to wrap it up in the next.
Oh no, yeah, sorry about that. So these are just showing the plates. Unfortunately, we did not find any zones of inhibition. So that leads just to future directions about like the biology process and things. But yes, that's it. Okay, all right.
Great, thank you so much. And our last presenter, Iyinyeoluwa.
All right, I'm going to share my screen. Okay, hold on 1 second while I. Okay. All right, and I hope everybody can see that.
We can see it.
Okay, that's great. All right, well, good morning, everybody. My name is Iyinyeoluwa Ruth Okulate. I am a second-year biology major at the University of Maryland Eastern Shore. Today, I will be presenting my project on the functional characterization of NHE3 and insulin in the zebrafish kidney. So maintaining body fluid osmotic homeostasis is a very vital characteristic of almost every living organism or every living organism. Sodium ion and water balance, maintenance, and regulation are key to maintaining this body fluid osmotic homeostasis in these organisms. And this process of sodium ion, water balance, and regulation is dependent on regulated transport across polarized epithelial cell membranes. So one of the most important proteins in the proximal tubule of the kidney, which is very much responsible for the majority of sodium water reabsorption and plays a key role in maintaining body fluid and osmotic homeostasis, is the sodium proton exchanger, NHE3. So NHE3 achieves this by achieves sodium water reabsorption in the proximal tubule of the kidney by utilizing an inward sodium ion concentration gradient generated by the sodium potassium ATPase to extrude hydrogen ions against this electrochemical gradient, as shown in this image right here. So since NHE3A is so important in the reabsorption of sodium ions in water in the proximal tubule, such small changes in its activity can have a relatively large impact on biological functions, biological processes, homeostasis, different things going on in our body, such as our blood pressure, our blood volume, etc. So an increase in the NHE3A activity can lead to a large increase in blood pressure. Can lead to a large increase in blood pressure and blood volume, and also a decrease in NHE3 activity can lead to a large drop that could be harmful in blood pressure and blood volume. So it's already well known, and it's been studied that insulin signaling regulates blood glucose levels. However, previous studies have also suggested that insulin signaling plays a role in proximal tubule transport through a stimulatory effect on NHE3A through through expression and activity, as shown in this diagram here. The stimulatory effect on NHE3 activity means that insulin could lead to an increase or a decrease in NHE3 activity, which, as previously discussed, could have large and important impact on body functions and processes such as blood volume and blood pressure. However, the signaling pathways and the signaling mechanisms that link insulin to the stimulation of an increase in NHE3 through activity, through proximal tubule transport still remain unclear. So that's what my project focused on, and the goal of my study was to determine the effects of insulin on NHE3 expression using zebrafish as a model organism. So you might be wondering why I use zebrafish as a model organism. We use the zebrafish as a model organism because they're cost effective, they're small in size, easy to maintain and breed in the lab, and they have structural and physiological similarity between the mammal and the zebrafish kidney, which means that the zebrafish kidneys are homologous to those of humans and mammals, as shown here. The structures do have a lot of similarity. So to conduct this research, adult zebrafish were divided randomly into groups, which each had 5 fish per group. They were injected intraperitoneally with 10 microliters of saline or insulin in 3 concentrations, 0.01 units, 0.1 unit, and 1 unit per kilogram. So the average fish weight is shown right here. And if you look over here, you can see the intraperitoneal injection taking place. So to do the intraperitoneal injection, the fish were first anesthetized and then injected, and then they had a 30-minute recovery period, after which they were dissected. Now, the insulin that was used for the injection was prepared by serial dilution of insulin to have the 3 concentrations listed here. The last group was injected with saline solution for control. Okay, so after the 30 minutes, they were euthanized. And right here, as you can see the dissection process going on. The kidneys were dissected and they were prepared for gene expression analysis by PCR and gel electrophoresis. So if you look at the video right here, which is almost over, the kidney is currently being pulled out, being excised from the zebrafish. Okay, so I also extracted and dissected the brains of the zebrafish to see if there was any effect on the expression of NHE3 on the brains, which not much is known about that. So the dissected kidneys were pulled together and they were homogenized in Trizol. They were purified to extract the total RNA as seen in this image here. And then using a spectrophotometer, seen here as using a spectrophotometer, I measured the RNA concentration to determine the volume of 1 microgram of RNA necessary to make up the complementary DNA and to synthesize complementary DNA using the thermocycler. After the complementary DNA were synthesized, I separated these into several tubes, about 36 tubes roughly, involving 7 tubes for the loading control, which is the Z-beta-actin loading control, and another 29 for the different primers for NHE3 through the different primers that were going to be used to analyze this. So after those were prepared, they were put in the thermocycler for the PCR to begin. And then after the PCR process was completed, that was all loaded for the gel electrophoresis. After that was completed, then the results were obtained and then processed. And so these were the results that we obtained from this experiment that we did. Our results showed that normalized by beta-actin, NHE3 expression increased slightly. From NHE3 expression increased slightly as the insulin concentration increased from 0.01 to 1 unit per kilogram, also seen right here. It also showed a similar expression of activity in NHE3 from smaller, this is NHE3B, from smaller concentrations of insulin to larger concentration of insulin in the zebrafish kidneys. And then when we move on to the brain of the zebrafish, we could see that there was also an increased concentration, slightly with the increased expression of both NHE3A and NHE3B with increased insulin concentration injected into the fish. So for the data obtained for the zebrafish kidneys, this is consistent with data that has been that showed that the increase in insulin would lead to a subsequent increase in expression or increased activity of NHE3 in zebrafish kidneys. In the brain, we did see a decrease shown between the 0.01 unit per kilogram and the 0.1 unit per kilogram normalized by beta-actin. And then it does increase again, but this will call for further investigation since not much is known about the effects of NHE3, the effects of insulin on NHE3 in the brain. But overall, the findings were consistent with and able to show the increase in insulin concentration in the proximal tubule of the kidney and in the brain as well will increase NHE3 expression in zebrafish. So gene expression analysis conducted using PCR revealed that the insulin injection stimulated mRNA expression of NHE3A and NHE3B. This data does suggest a link between insulin and NHE3 in activity in the proximal tubule of the kidney, and it provides insight into how high blood pressure or hypertension may develop in patients with type 2 diabetes or hyperinsulinemia. As discussed previously, insulin may lead to a stimulatory effect on any NHE3A activity or NHE3 activity that can have a large impact on blood volume and blood pressure. And so this data also suggested a link between insulin and NHE3 activity in the brain, but as mentioned before, this will call for further investigation since we don't really know much on this and this hasn't really been studied much. And so for future studies, which are currently ongoing, we are increasing the number of biological replicates per group. And we intend to perform the qPCR studies to determine the gene expression of NHE3A, NHE3B insulin receptor A, insulin receptor B, normalized by beta-actin, which is the loading control. And we also intend to perform this study to test different concentrations of insulin for the IP injection besides the 1 unit per kilogram, 0.01 and 0.1 units that were tested in this first part of the study. Everyone at UMES that has afforded me the opportunity to carry out this research. Thank you.
Thank you so much. Great. Well, wow, I'm so impressed with all four of you. Very different aspects there of genetics and biochemistry, but I think what really came across was your passion for these projects. There was a question earlier in the chat that I'd like to open up to all four of you in terms of, do you have an interest in continuing this line of research? And what in general from from your research have you learned about kind of the overall research process? I'm going to open that up to all four of you.
Yeah, I guess I can start. I would be potentially interested in continuing this line of research. I am a graduating senior, and so my goal is to become a physician, but I have a couple gap years or whatever before I do that. So I'm definitely interested in continuing research. I'm not sure if it will be on this same project or not, but overall from this, I've done a few research projects throughout my college career, and from this one, I think it just shows that like anything can happen, and like the expected is not going to happen ever, like usually ever. And even so, like you just learn to adapt with all the new challenges you're facing. I think that's kind of been the motto for the year.
Great, thank you.
I can go next. So I'm also a graduating senior, so I probably won't continue this exact project at my university. However, I'm definitely interested in continuing along a similar line because I'm very passionate about dairy cattle genetics, especially as a way to improve animal health and welfare, which is why I chose this area for hornless genetics, because it's highly related to that. So I definitely would like to either continue on the polled end or in other areas of dairy cattle health and welfare genetics. And as far as what I've learned, that if there's a roadblock, it will be thrown at you. Definitely. This year has definitely been very challenging, and this project has taught me how to adapt to different situations and to always find a plan B and have a plan C just in case.
Great, thank you.
I'll go next.
Okay.
Oh, sorry.
All right. I would say for me, because I know everyone else is the two people who just spoke before me were graduating seniors, but I'm a second year. So this was kind of my project that I did, like my first project in undergrad. So for me, I definitely would like to continue this, especially because the big picture behind why I'm doing this project is really important to me. Hypertension and hyperinsulinemia they're two things that have affected my family personally. And I didn't even, so that wasn't on my mind when I picked this project, but for me, I really have, fallen in love with doing this research into this project, and I want to keep going to see more, which is why we're still doing more on it. But yeah, I think from what I could say I've learned from the almost 2 years of research experience I have now is just you have to be willing to grow, and you have to be willing to, just find new ways to do things, especially in the pandemic. We all had to find new ways to do things. You just have to be willing to adapt. And I think it's been a really good experience for me.
Fantastic.
And yeah, I would definitely have to agree with all of what everyone has been saying so far, especially about what things that you've learned and things, because it's really just, especially when it comes to research, it's just about adapting and finding new ways to do things. I'm a junior, so I have another more year of research to go. So I don't think I'll be doing this particular study, but I know I'll be doing something similar that deals along with the same idea of kind of like medicinal chemistry, which is something I've really enjoyed and really liked. And even after the future, I'm planning on going into research that deals with chemistry and then hopefully going into like medicinal chemistry and stuff. So stuff like this, I always think it's super interesting to kind of look at like how they kind of make this stuff and then how it's used by doctors and in people all the time and stuff. So it's super interesting and something to really look at a lot.
Great. Any other questions from our audience? No? Well, I think you all did a fantastic job. You should really be proud of yourselves, not only for your presentation, but the research, very impressive. And it's just interesting also to see the diversity of, the ways that this kind of research can be adapted to different kinds of questions. So really, really interesting to see that. Thank you for your presentations. And we have our closing ceremony in a little under an hour. So hoping to see you all there, and enjoy the rest of your day. And congratulations, you all did a wonderful job.
Thank you. Great job, everyone.
Thank you.
Thank
Nice
you.
seeing you all.
Take care.